Obtain the mesh file and also the full training movie by purchasing this product.
In this project, we will simulate hyperthermia therapy of a biological tissue which is involved with a cancer tumor. Common methods of cancer treatment include surgery, radiotherapy, and chemotherapy. These methods have disadvantages including aggressiveness, irreversibility, and side effects. Warming in hypothermia therapy prevents oxygen and nutrients from getting into the unhealthy tissue. In this way, proteins within the tissue change nature and can surround the cancer cells. This makes it possible for the immune system to identify cancer cells and destroy them. The present study examined blood flow in capillaries passing through a tissue containing cancerous tumors and using hypothermia therapy. For this purpose, we assume a spherical space to be an example of a healthy body tissue or cell in which blood flows at very slow rate. There are several veins within this tissue. The vein structure of cells and tissues of the body are like bushes. To simplify the problem, we assume that all the veins line uniformly in the tissue, which is in x-axis. As the blood flow passes through the spaces within the tissue, the whole tissue can be considered as a porous medium. There are 4 spherical cancerous tumors of different diameters at the center of this tissue and on the surface of the veins as biological tissue. In fact, the thermophysical properties of healthy tissue differ from those of cancer cells. But to simplify this, we assume the thermophysical properties of the healthy tissue similar to the unhealthy cells. Every cancerous tissue is a heat source with heat energy per unit of volume to cause heat transfer and thereby significantly increase blood flow. The purpose of this study is to investigate the response of blood vessels and body tissues to this heat. The geometry of the present model is designed in ANSYS Design Modeler software. The present model comprises of a cylindrical tube as the capillary, 4 spherical blood vessels attached to the capillary as tumors, and a spherical space for intra-arterial blood flow around the vein, which has a slower flow rate than flow inside the capillaries. The spherical tissues around the capillary have a different radius. The outer sphere is also considered as the space for blood flow within the tissue and around the capillary, which contains 2 input and output hemispheres. The meshing of the present model is performed by ANSYS meshing software, and the present grid type is unstructured, and the total element number is equal to 770,087. As was previously explained in the blood material, by clicking on tissue material, again a new window will appear in which you are able to change the settings for this material under the properties section. Now, as for the cell zone condition, 2 sections of fluid and solid zone condition are defined. Under the fluid section, you will see that 2 zones of part artery and part big are defined. By clicking on part artery, which is related to the arterial vessels, after clicking on it, you will see that a new window will appear in which we define the material named blood for this zone. Now, as for the solid zones, starting from part 3 to part 7, these parts refers to the different cancer cells we have designed in our geometry. The settings for all these zones are like each other, and by clicking on, for example, part 3, a new window will appear which lets us to change the settings for these zones. You will see that an energy source term is defined. By clicking on edit, we will be able to change this source term. For example, under the momentum tab, you can see that the velocity is selected to be in coordinate system of Cartesian, which you can see that x velocity is defined as the value indicated, and under the thermal tab you will be able to see the temperature of this flow entering this boundary. Now, under the outlet section, the artery outlet, which is related to the blood vein outlet, as you can see, it is defined in this place. You can see that the type of this boundary is set to be pressure outlet. Again, by clicking on edit, you will be able to change the settings for this boundary. In the appeared window, as you can see, under the wall motion section, the stationary wall is defined and the shear condition is defined to be no sleep. By clicking on thermal, you will be able to see the thermal settings for this boundary. After double-clicking on the initialization button, a new window will appear showing you different methods of initialization, hybrid and standard. Now in the standard initialization method, you get to choose the first amounts and values for the first iteration of the simulation progress. These values refers to the values used in the first iteration of the simulation progress, and if you choose the values for each parameter wisely, your simulation progress will finish sooner. It should be mentioned that you can also choose the first values or the initial values by just clicking on the compute from drop-down list and clicking on one boundary. For example, by clicking on compute from all zones, the software will automatically average the values in different zones and boundaries and put those values in the initial values for the software. Now, as you can see, and as was explained in the previous parts, we have defined a heat generation rate inside the cancerous cells in order to simulate the hyperthermia process. As a result of that, you can see the increased temperature of these cancer cells, which is clearly shown in this contour. Now, in order to extract a 2D temperature contour, we first have to define a plane on our geometry. To do this, you just have to click on location and then select the plane and accept the appeared window. Again, on the low left side of the software window, you can see under the definition section actually, in front of the method, you can see that we have defined our plane to be as an XY plane with the value of z equal to 0. And by clicking on apply, you will see that a plane, just like the picture you can see right now, is defined in your software. Now, in order to define a 2D temperature contour and put that contour on the defined plane, you just have to click on 2D contours and then accept the appeared window. On the low left side of the software, again in front of the location, just have to select the defined plane, which is the plane we have defined in the previous slides, and the name of that plane is plane 1. After that, we just have to select the variable, which in this state is temperature. After that, just by clicking on apply, you will be able to see the temperature contour defined on that plane. Finally, a summary of different settings and setup that we have used in our project is presented to you in the slide.
Obtain the mesh file and also the full training movie by purchasing this product. To benefit from MR-CFD services including simulation, consultation, and training, contact our experts via info@mrcfd.com. Hope you enjoy.